Welcome to the special session entitled SARS-CoV-2 and Wildlife Health. I am David McNaughton, a Natural Resources Specialist for the Navy at NAVFAC Southwest, and I am co-chairing this session with Katie Heyman, a wildlife veterinarian for Washington Department of Fish and Wildlife. Our speakers will take up the full time, so we will take questions down in the chat. Please raise your hand so Katie and I can cue your question and the speakers can answer. I apologize in advance if we miss any of your questions, but feel free to contact either of us after the session and we can connect you to the presenters. Our contact info is on this slide.
So, why are we here today? The speakers in this special session will cover several key topics related to SARS-CoV-2 in North American wildlife. We'll start out by providing an overview of the state of knowledge of SARS-CoV-2 in wildlife, and then we'll discuss a strategy and policy to combat wild SARS-CoV-2 reservoirs. We'll also highlight known reservoirs and zoonotic paths from recent findings, and then wrap up with a discussion of ongoing research that pertains to the susceptibility of North American wildlife. Attendees of this session should walk away with a solid understanding of the risks associated with SARS-CoV-2 that are faced by North American wildlife. As a zoonotic disease, SARS-CoV-2 is quickly becoming a poster child for One Health, with key news headlines highlighting just this fact. From tigers and lions and other big cats at several zoo facilities across North America to the gorillas at the San Diego Zoo, the possible impacts of this virus on wild animal health has been exemplified. Mink and mink farms are no exception, and perhaps are even more important given the rapid transmission of SARS-CoV-2 to mink from humans in North America and in Europe, and in some cases in Europe back to humans from the mink. These headlines and the stories behind them are why we are here today.
I would like to introduce you to our speakers and topics for this session. Dr. Patty Klein is the U.S. Forest Service National Program Lead for Fish and Wildlife Health and the attending veterinarian for the Forest Service IACUC. She will be discussing SARS-CoV-2 strategy and policy today. Dr. Jonathan Sleeman is the Center Director for the USGS National Wildlife Health Center. He will be highlighting current approaches and research related to SARS-CoV-2 at the center. Dr. Jana Mazet is a professor of epidemiology and disease ecology in the UC Davis One Health Institute and the School of Veterinary Medicine. She will describe the One Health strategy and program at UC Davis and some of their relevant recent findings. Dr. Susan Schreiner is a research biologist at the USDA National Wildlife Research Center on the National Wildlife Disease Program. She will be presenting on recent outbreaks in wild fur bearers and fur industry reservoirs for SARS-CoV-2 and COVID-19. Dr. Jonathan Cook is a postdoctoral research associate at the Patuxent Wildlife Research Center. He will re-examine the USGS bat reservoir risk analysis published last May and update us with new data and lessons learned. Finally, Dr. Mitch Palmer is a research scientist for the USDA at National Animal Disease Center. He will demonstrate his findings on white-tailed deer susceptibility to the virus. Now please enjoy the session. Remember, we will be answering questions live in the chat. See you there.
Hello, I'm Dr. Pat Klein, the National Program Lead for Fish and Wildlife Health at the USDA Forest Service, and I'm pleased to be with you here today. As we continue to navigate through the risks and challenges of the COVID-19 pandemic, I hope this presentation offers some useful perspective on One Health coordination for SARS-CoV-2 risk mitigation in animals and people, and these are built on lessons learned from previous emerging disease outbreak experiences. Well, I'm going to go ahead and turn off my camera, and then I'll pop up at the other end. Meanwhile, we'll go through the slides together. Thanks. Let's consider the challenges and opportunities for state and federal agencies and our partners to prepare for and respond to new disease events. There are many factors that influence disease emergence or expansion. One key factor or challenge is the increasing pathogen exposure risk at the wildlife, domestic animal, human population interface. Well, what do we mean by interface or interface diseases? These can be endemic, Diseases that are already present in an area that may further expand. They can be foreign diseases or introduced diseases, or they can be zoonotic. They're also promoted by changes in climate or changes in environmental circumstances such as drought or fire or hurricane, and anthropogenic events such as international travel. There's always a critical point at which cross-species disease transmission occurs. Now, while there are always challenges, there are also opportunities. We have opportunities to interrupt disease transmission at the animal-human population interface. We also can develop structured decision-making processes to help mitigate disease risk, and we can create effective cooperative partnerships for a One Health response. In this presentation, please keep this diagram in mind. Please see how the human health, animal health, and environmental health are interconnected, and all the disciplines involved in understanding those relationships. At the center is the One Health concept, which recognizes there are no dividing lines. Rather, there is an interdependence between all components. And as Rudolf Virchow said, between animal and human medicine, there are no dividing lines, nor should there be. Another way to look at the One Health epidemiological triad is depicted here, showing wildlife, domestic animal, and human populations intersecting, surrounded by a common shared environment or ecosystem. We all breathe the same air, we use the same water sources, and we rely on the landscape and suitable habitat. Disease results from dynamic interactions of the pathogenic agent, an animal or human host, and the environment, which are always changing. Therefore, emerging diseases are the norm rather than the exception, and interface diseases are increasingly common. Many people have referred to and quoted from this Kate Jones article published back in 2008 that described global trends in emerging infectious diseases, or EIDs, and correlations to zoonotic diseases originating in wildlife. EIDs are a significant burden on global economies and public health. In this report, they analyzed over 300 emerging infectious disease events over a 60-year period, and they showed how the EIDs rose significantly over time and were dominated by zoonotic diseases. In this case, it was over 60% of those diseases were zoonotic, and the majority of those, more than 70%, originated in wildlife. Also, at least 50% or more of those were caused by bacteria or other microbes, such as rickettsia, that had antimicrobial resistance concerns. These EIDs are significantly correlated with socioeconomic, environmental, and ecological factors and provide a basis for identifying global regions as emerging disease hotspots. Their conclusion was that the global resources at that time were being poorly allocated, and that really surveillance efforts should be focused on countries or areas where the next important emerging infectious disease is likely to originate. Historically, avian influenza outbreaks, especially the H5N1 high-path avian influenza outbreak, created a turning point in the movement towards a One Health strategic response. This diagram represents the complexity and versatility of avian influenza viruses to mutate and adapt to many host species. Certain wild birds, such as waterfowl or shorebirds, are reservoirs for avian influenza viruses that can spill over to infect other species if and when circumstances permit. The H5N1 poultry outbreaks and human case reports in China and Southeast Asia back in 2003 prompted the U.S. government to develop comprehensive all-government H5N1 playbooks and pandemic response plans identifying interagency coordination. Interagency coordination included USDA focusing on poultry and wild bird surveillance, on animal vaccine development, and even banning live poultry importation from affected countries. The USGS, for example, focused on wild bird surveillance and wildlife mortality investigations. The CDC emphasized public health preparedness and pandemic plans, and also PPE guidance. And FDA was focused on human vaccines or antiviral drug development, and even banned importation of poultry products and eggs from affected countries. The outcome was that H5N1 never was introduced into the United States. Also important to remember that highly pathogenic avian influenza is a reportable disease per USDA regulations. So there are and were existing surveillance and response plans in place for these types of foreign animal diseases. That's known as the USDA Red Book, and those plans are periodically updated. These plans describe interagency coordination and in preparedness and response. There are also the overarching federal laws, such as the Animal Health Protection Act, which USDA has authority to protect livestock and poultry, stop animal movement, etc. And CDC and FDA have authority under the Public Health Services Act to protect public health from communicable diseases. U.S. Fish and Wildlife Service, of course, has authority under the Migratory Bird Treaty Act, Endangered Species Act, Lacey Act, etc., to regulate wild bird movement, wildlife trade, transport, etc. Moreover, that H5N1 epidemic in Europe, Africa, and Asia also prompted the U.S. government to develop interagency strategic plans for wild bird surveillance, and also a North American plan for animal and pandemic influenza with our partners in Canada and Mexico. A One Health disease response strategy was developed, and we were prepared. And this North American plan could be a template for future pandemics to be modified accordingly. So, how do these plans prepare us for the next emerging zoonotic disease threat? Can we use these One Health lessons learned from the H5N1 response to prepare for future events? And the answer is yes. So similar to avian influenza viruses as a large virus family that continually reassorts and mutates, coronaviruses also are a very large viral family divided into 4 major groups, and they're also known to obviously mutate and evolve over time. We have the alpha, beta, gamma, and delta coronaviruses. Where the alpha and the beta coronaviruses infect mammals, including humans. We have examples of that with feline infectious peritonitis in cats. We have enteric diseases in dogs and cows, and even in pigs with transmissible gastroenteritis. And there are many types of human coronaviruses known as the common cold viruses that have existed for quite some time. The gamma and delta viruses actually infect avian species, mostly fowl such as poultry, infectious bronchitis virus, for example. But let's focus on the beta coronaviruses to review the emergence of SARS and MERS that evolved from a bat reservoir host. In 2002, we experienced how rapidly a newly emerging zoonotic disease can spread globally through international travel, and we also realized the connection to live animal markets as a mixing bowl of animal infections that can evolve to infect humans. The use of genetic fingerprinting and genomic analysis, comparing gene sequences of virus strains, were relatively new techniques at that time, but revealed epidemiological evidence suggesting the animal origin of the SARS virus. And subsequently, this was linked to the potential intermediate host, the palm civets, which were being sold in those markets, and subsequently linked to the potential horseshoe bat reservoir in virus evolution. This was a remarkable One Health epidemiological investigation that established the use of genetic analysis as integral part of the epi process. This diagram does summarize how SARS and MERS likely spilled into intermediate mammalian hosts and further evolved to become infectious to humans. But, this doesn't happen overnight. We still don't know the timelines for these coronavirus mutations in other animals, nor the timeline for adaptation for human-to-human transmission. And of course, in 2019, another novel beta coronavirus, SARS-CoV-2, evolved, which has resulted in a global pandemic and an emerging zoonotic disease. So One Health coordination to identify and mitigate these risks has been essential. Well, how do we go about doing this? Well, we do it together, and we do it synergistically. Given these past experiences with other emerging zoonotic diseases, the One Health community immediately recognized the essential value of the interagency collaboration and coordination. The CDC One Health Office began hosting weekly interagency conference calls early in the COVID-19 pandemic, especially as the first reports of SARS-CoV-2 positive pet dogs in China from their infected owners, and later on pet cats in Europe and Asia, and other infected animals became known. The One Health Federal Interagency COVID-19 Coordination Group, or OFIC abbreviated, has grown to over 20 federal agencies and more than 130 U.S. government and non-government partners. This is a direct outcome of the H5N1 period when the concept of One Health and bringing public health, animal health, and environmental health professionals together was novel. Fortunately, now it's becoming routine. One immediate advantage to having this One Health consortium of scientists and experts is just-in-time science. This slide represents a recent synopsis of experimental and natural infections in different animals that help to guide us in our decision-making. You can see in the green circles, these are animals considered highly susceptible. Natural infections in big cats, such as the lions and tigers and pumas at zoos. Obviously pet cats from infected owners. We've had multiple outbreaks now in farmed mink. We'll talk about that in a second. And even recently we've had non-human primates such as gorillas at the San Diego Wild Animal Park. There was an experimental study recently by the Agricultural Research Service in white-tailed deer, which did document that experimentally infected white-tailed deer can develop upper respiratory infections and transmit that to non-inoculated cohorts. The species in yellow are considered to be moderately susceptible, such as natural infections in pet dogs from their infected owners, and the animals listed here in pink are presumed to be not susceptible at this time, including some North American bats, and there are other speakers in this special session that will speak more specifically to that. So as of mid-January, when this slide was prepared, we had then 16 mink farms in 4 different states, Utah, Wisconsin, Michigan, and Oregon, that had reported SARS-CoV-2 outbreaks in their farmed mink and also in their farm workers. Many of the mink farms had actually over a 50% mink mortality rate. The CDC, APHIS, USGS, and the respective state agencies, the agricultural, wildlife, and public health departments, are all coordinating the mink farm epi investigation. One wild mink captured in proximity to a positive mink farm in Utah actually tested SARS-CoV-2 positive, raising concerns for spillover into native wildlife. And as listed here on the slide, there's a very, very new, recently, early release article in the CDC Emerging Infectious Disease Journal that describes serosurveillance results with high SARS-CoV-2 antibody titers in free-ranging, presumably escaped farm mink, suggesting an infection pathway to native wildlife. You can access that article by the web link that's provided on the bottom of the slide. So interagency coordination of SARS-CoV-2 surveillance continues of feral or escaped farm mink, of wild mink, and other animals surrounding these mink farms. Some of the wild animals were radio collared to follow their movement in proximity to the farms. However, it must be noted that at this time, routine testing of animals is not recommended. However, there have been criteria established for testing of North American wildlife, as you can see here on the slide. And if testing is determined in consultation with both federal and state wildlife health, domestic animal, and public health officials, wildlife health specialists should submit samples and should communicate this relevant information to those various state and federal partners. Remember, any SARS-CoV-2 test that is presumptive positive at a local or state diagnostic lab must be confirmed by NVSL, because NVSL is the official reporting entity in the United States to report positive results to the OIE. And again, please reference those web links at the bottom of the slide for additional information. This slide also is a synopsis of what we know at this time of what at-risk wildlife are indicated as high risk or relative risk or possible risk. This is based on experimental and natural infections, also several susceptibility modeling studies using ACE2 receptors. There are several risk assessments as well that have been developed, and there have been a few infectivity studies. Of course, this list is subject to change as we continue to learn more. I would like to at least acknowledge, too, that even the Association of Fish and Wildlife Agencies and USGS have developed some initial guidance statements on, for example, COVID-19 and bat-related activities. That was developed back in April of last year in coordination with the Bat Working Group and the AFO Fish and Wildlife Health Committee. Later on, in June of last year, there also was an additional statement put out about North American species of mustelids, felids, and canids relative to COVID-19. And again, a rapid risk assessment has been completed by the USGS folks. Many federal and state agencies have developed their own guidelines based on the known science and risk factors and with intention to be consistent with their partners in that region. I'd like to spend a few minutes talking about the hierarchy of controls, which was developed by the CDC, National Institute of Occupational Safety and Health, as a risk assessment tool for structured decision-making on mitigating risks from various hazards such as biological, chemical, or physical for overall health and safety. The CDC OFIC Wildlife and Zoo Subgroup has adopted this hierarchy of control tool to help guide wildlife professionals, scientists, managers, and wildlife rehabilitators to reduce the risk of SARS-CoV-2 transmission to and between people and animals. This diagram depicts how the controls are ordered from least effective to most effective. Note that PPE is considered to be least effective because it can fail if not used properly. not changing disposable gloves between patients, or not getting fit tested for certain types of respirators such as N95s. Also, PPE is really good, but is best if it's used in combination with administrative and engineering controls. The only way to completely reduce a risk to zero is really to eliminate that risk or the hazard. That would mean stopping any work with at-risk animals. However, we feel that we can use this hierarchy of control tool and its components to allow wildlife work to continue, provided the risk can be minimized by incorporating PPE, administrative, and engineering controls. Examples would include temporary suspension or postponement of direct contact with wild animals, reassuring or ensuring that proper use of PPE and disinfectants are used when handling animals and in between, develop animal handling protocols to minimize disease transmission potential, and also the importance of personal self-monitoring and testing for SARS-CoV-2 if individuals feel that they are symptomatic. The CDC OFIC has developed these risk mitigation guidelines using the hierarchy of control tool and has posted that information on the CDC COVID-19 website, and that web link is posted here or provided here at the bottom of the slide. Also, the potential for reverse zoonosis, in other words, infected humans transmitting SARS-CoV-2 back to animals or spillback, has been reported in pets and captive wildlife. But these concerns still extend to our North American wildlife that could be exposed by infected humans through direct contact or in close proximity, such as recreational activities like caving, or in the capture or handling of animals in field research, or during wildlife rehabilitation activities. So in order to address these concerns, the International Union for Conservation of Nature, or IUCN, developed guidelines on preventive measures to reduce the risk of reverse zoonosis. The diagram depicted here on the left of the slide is showing what the exposure risks could be as direct contact or aerosol or environmental exposure, and some of the mitigation strategies to minimize or assess and protect against transmission. Also, there's a really good case study on bats that was published by Kevin Olival and his co-authors on the risk of reverse zoonotic transmission of SARS-CoV-2 to free-ranging wildlife, and that article can be accessed through the information on the slide. The consequences for reverse zoonosis could be actual wildlife mortality, risk to conservation efforts, and infection of threatened and endangered species. Also, it could provide for disease spread and establishment of wildlife reservoirs. So we really need to pay attention to preventing reverse zoonoses. There have been other efforts too to look at the sort of big picture of these types of emerging diseases and developing risk mitigation strategies. In 2017, the CDC, USDA, and DOI organized a One Health Zoonotic Disease Prioritization Workshop to further the efforts to address zoonotic disease challenges in the United States. And during the workshop, the participants identified a list of relevant zoonotic diseases and prioritized them. The 8 top zoonotic diseases are listed here on the slide. Interesting, the zoonotic influenzas and obviously emerging coronaviruses were already on that list. Even West Nile virus, which continues to still affect even ruffed grouse and other pheasant populations and other wild birds and wild animals in the United States. The goal of the workshop was to establish a framework to then further help develop plans for implementing and strengthening these multi-sectoral One Health approaches to addressing these diseases in the United States. In 2018, the World Bank actually developed an operational framework for strengthening human, animal, and environmental health systems at their interface. They realized the socioeconomic costs of emerging and pandemic diseases, and that these diseases will continue to increase given the wide-scale changes in land use, transformation of agricultural practices without adequate biosecurity, climate changes, weather changes, trade, travel, urbanization, all these different components that would promote these types of diseases. One of the emphasis that they made, a key emphasis in the framework document, is to target these disease threats upstream. In other words, look at preventing at the source. Oftentimes that is a wildlife reservoir, animal reservoir, but look at those situations before spillover can occur, spillover into perhaps domestic animals and even then amplified in the human population. The National Academies of Science, Engineering, and Medicine is actually hosting a workshop February 23rd to the 25th on systematizing the One Health approach in preparedness and response efforts for infectious disease outbreaks. Hopefully we'll be able to provide some highlights from this workshop during our special session Q&A. Also, in the previous Congress, 116th Congress, several proposed legislative bills were introduced. Unfortunately, none of them were actually succeeded, but we're hoping that perhaps in the new Congress they might be reintroduced. One bill required Health and Human Services and USDA, in coordination with other specified agencies and departments, to develop a national One Health framework for coordinated federal activities to better prevent and prepare and respond to zoonotic disease outbreaks, and to advance our understanding of the connections between animal, human, and environmental health. This was introduced in both the House and the Senate, and that was called the Advancing Emergency Preparedness Through One Health Act. Another proposed legislation in the previous Congress was also introduced in both the House and Senate, and that was called Preventing Future Pandemics Act. That would have authorized the U.S. government agencies to crack down on international wildlife markets and global wildlife trade for human consumption. So what are the lessons learned now? What has worked well so far? Well, we really do have ongoing and improving One Health collaboration and integration for disease preparedness and response. We are developing very excellent risk assessments to help facilitate strategic risk mitigation decisions. We're coordinating our animal and human health surveillance systems, and we're improving our diagnostic lab capacity and networks. We're also making sure that we establish good communication channels with timely reporting mechanisms. But there are always gaps. There are always the need for advances in scientific information. We still need to learn more about susceptibility of animal and human hosts, the extent of pathogen range and virulence, the dynamics of interspecies transmission, and the effect of the environment on disease transmission. We also need validated test methods for testing multiple different species of animals and different samples. We especially need legislated resources to assist us in disease prevention and response. So the recommendations really are to continue to bridge these gaps, to recognize the key roles and authorities of the respective agencies, and to continue to strengthen these collaborative interagency One Health partnerships for disease preparedness and response. We really need to engage everyone: scientists, diagnosticians, the animal and public health specialists, epidemiologists, biologists, ecologists, land managers, decision makers, legislators, and to meet these future disease challenges. We really need to continue to promote our public-private partnerships, and working together really does work. Thank you very much, and I look forward to talking with you soon. Have a great day.
Hello, my name is Jonathan Sleeman. I'm the Center Director with the U.S. Geological Survey's National Wildlife Health Center, and appreciate the opportunity to spend the next few minutes to discuss our scientific response to the COVID-19 pandemic. I'm going to talk very briefly about the origins of SARS-CoV-2, talk a little bit about the risks, or potential risks to wildlife, and the various scientific methodologies we've been applying to understand, characterize, and manage these risks. So whilst the origins of SARS-CoV-2 have still not been fully determined, it's highly likely that it's an example of pathogen spillover. There are a wide variety of different pathogens, particularly viruses, that are found in wildlife reservoirs or hosts, including rodents, bats, and so forth. There are certain human activities or human behaviors that result in high-risk human-wildlife interactions, such as land use change that's bringing wildlife into close contact with people, or through the trade and consumption of wildlife, as shown in this picture. Here are fruit bats being traded in a market in China. These viruses can then infect people. They can mutate and evolve to efficiently transmit from person to person, and then they can become global pandemics because of the rapid transportation of these viruses through global travel. Now, wildlife are natural reservoirs for different varieties of coronaviruses. And coronaviruses are interesting because they're enveloped positive sense single-stranded RNA viruses, which makes them prone to copy edits on replication or reproduction, which means they readily mutate and readily adapt to new species. Now, they're divided into 4 different genera, The alpha coronaviruses are found primarily in mammals, including bats. The beta coronaviruses are also found in a wide range of mammals. And this is the group of particular interest because since 2003, there have been now 3 coronaviruses, beta coronaviruses, that have emerged from wildlife that have infected humans and caused epidemics or pandemics. This includes SARS back in 2003, which is thought to originate from bats and transmitted via palm civets, the Middle Eastern respiratory syndrome associated with camels, and now the coronavirus disease, or COVID-19. The gamma and the delta coronaviruses are carried primarily by avian hosts and are not known to pose a disease risk to humans. Now, this image just shows you the wide variety of different coronaviruses that are found in a wide variety of different species. Obviously, a lot associated with different bat species, but also there's those found in felids, in rodents, in also unusual species like beluga whales and so forth. So there's a huge variety of different coronaviruses. So obviously, the logical question to ask is if these viruses or this SARS-CoV-2 did originate from wildlife species, and wildlife can host a wide range of different coronaviruses, what are the potential risks to wildlife of this virus getting back into wildlife populations? And there's been some very interesting studies looking at the sort of homology of the ACE2 receptor, which is the main cell receptor by which the spike protein of the SARS-CoV-2 binds to infect a cell. Looking at the genetic relatedness of the ACE2 receptor of a variety of species. And from these studies, there's been a wide range of species predicted to be susceptible to SARS-CoV-2. Obviously includes the great apes and primates, but also interestingly white-tailed deer, Chinese hamsters, anteaters, and even marine mammals. And so this has led to a lot of questions being raised by the wildlife management community, whether these viruses do potentially pose a risk. So just taking a little step back, managing emerging diseases is particularly challenging because there's lots of knowledge gaps and lots of unknowns. And there are a variety of different things that we need to do in order to understand and respond scientifically to emerging diseases. We need to understand, identify the particular pathogen or hazard. We then need to characterize the hazard. We then need to develop diagnostic tests to identify and detect the hazard. We need to apply that diagnostic test to conduct surveillance in a variety of at-risk populations. We also need to develop different management tools. And whilst in my graphic here, I show it as a linear process, very often all these things can happen at the same time. So what I'm saying really is you need to mobilize a wide range of different techniques and methodologies in order to respond to emerging diseases. And we learned a lot from another disease associated with bats, which is bat white nose syndrome. This is caused by Pseudogenoascus destructans, a pathogen previously unknown to science. And so clearly, we refined our ability to respond to these diseases through understanding of white nose syndrome. Again, initially identifying the hazard, characterizing it, understanding why and how it kills bats, to the development of diagnostic test, to the conduction of conducting national level surveillance for white nose syndrome, and finally now with multiple entities trying to develop management tools, in our case including a vaccine. So back to SARS-CoV-2. So back in March of 2020, when this was first declared a pandemic of the World Health Organization, wildlife managers in the United States expressed concerns about the risks posed by SARS-CoV-2 to different bat populations. They really posed two different questions. Could it cause mortality? And could a novel reservoir be established in North America? And that had a third question about whether we could actually see mutation, evolution, and potentially recombination of SARS-CoV-2 with other coronaviruses, creating new strains. As a consequence of these challenges, many agencies issued interim guidance restricting various activities, including management and scientific activities, which obviously has a big impediment on the ability to conserve bat populations, particularly those impacted by white nose syndrome. So the first thing that we did was actually conduct a rapid risk assessment to try again really characterize the risks. What's the probability of these adverse events happening, like bats becoming infected by SARS-CoV-2 through human activities? And I'm not going to go into this in a lot more detail. Jonathan Cook is going to present the rapid risk assessments that we've conducted. But I just wanted to highlight the fact that these are very important tools to rapidly try and understand the probability of adverse events happening, as well as understand the uncertainty associated with these events. And there's very different methods that can be used depending on the amount of data that's available. You can do qualitative assessments, semi-quantitative assessments, as well as fully quantitative assessments. One of the best techniques to use to understand species susceptibility, the ability for a species to share and transmit a particular pathogen is through experimental challenge studies. So we were fortunate to have a colony of big brown bats at the National Wildlife Health Center that we've been using for white nose syndrome research. So we quickly repurposed some of this colony for an experimental challenge study of SARS-CoV-2. So we took 8 big brown bats. We inoculated them with a high dose of SARS-CoV-2 virus by the oral and nasal route, as shown by this picture. We then co-housed these eight bats in pairs with unexposed bats. And then we tracked these bats over a 3-week period to look for shedding by the oral and rectal route, also looking to see if there's a viral transmission from the exposed to the unexposed bats. And to summarize the results, we basically found no evidence of viral replication in any of the tissues of the exposed bats. We found no evidence of viral shedding from the swabs we collected for PCR analysis throughout the duration of the study. And we sacrificed all bats at the end of the study, and there's no evidence of any sign of infection or pathology in any of the tissues of these bats. So good news is, from this challenge study, this species in particular appears to be resistant to SARS-CoV-2 infection. Now, performing experimental trials and a whole range of different bat species obviously is challenging, it's expensive, it utilizes a lot of animals. And so one thing that we're trying with this particular virus is to do some in vitro experiments with bat species. So we've developed little brown bat keratinite cell lines. We're also working with the USDA's Agricultural Research Service to develop other cell lines that express the ACE2 receptors for different bat species. We can then infect these cell lines with the SARS-CoV-2 virus. We can look to see whether they're susceptible to infection. We actually can characterize the immune response of these cells. And one thing we're particularly interested in is testing the effects of co-infection of SARS-CoV-2 and white nose syndrome. There's some indication that infection with white nose syndrome may cause immunosuppression in the host and therefore leave them more susceptible to infection with coronaviruses and potentially higher shedding. So that's, I think, an important thing for us to look at. So this work is currently ongoing. Now, experimental studies will tell you whether a species is susceptible, but doesn't always translate into what is happening naturally in the field. And so we've been National Health Center in partnership with a whole range of state and federal wildlife management agencies conducts general scanning surveillance for all sorts of wildlife diseases across the country. And so through this established system of scanning surveillance, we've actually been opportunistically testing animals associated with mortality events for coronaviruses in general, as well as specifically for SARS-CoV-2. So we've actually implemented a pan-coronavirus PCR test to look for all coronaviruses. We've also utilizing the CDC's SARS-CoV-2 PCR test. And obviously we're focusing on what we believe are high-risk species, or species in high-risk situations, i.e. where they're in close contact with people. We've collected over about 1,000 samples so far from this surveillance system. I'm just going to show you here some preliminary results from surveillance in bats. As you can see, we've tested about 350 bats so far for coronaviruses. We have not found SARS-CoV-2. We have, though, however, found a wide variety of different alpha coronaviruses in these bat species. You can see the phylogeny of these different viruses that we've isolated or identified in the chart here. And we see about a prevalence of about 10% of coronaviruses in these different bat species. And obviously, it's very important to characterize naturally occurring coronaviruses in our native wildlife, for the very reason I mentioned earlier, is that we're obviously to see what are the risks for recombination or mutation of SARS-CoV-2. Now, obviously, since this pandemic, there's been more and more experimental studies and more sort of natural infections that have happened. We're beginning to understand the wide range of species that are potentially susceptible or have been demonstrated to be susceptible, particularly felids, as well as mustelids. And so outbreak investigations are also a very important part of the overall surveillance strategy. As you're probably aware, there's been large-scale outbreaks on mink farms, not just in the United States, but in other countries, particularly in Europe. These situations have a high risk of rapid viral transmission amongst the farm populations, as well as the spillover of the virus into animals that are surrounding these farms. And I'm not going to go into further detail because I believe Dr. Tom DiLiberto is going to talk in depth about the investigations at these mink farms and the wildlife mink farm interface, but just say that we have been participating in these One Health investigations of these mink farm outbreaks. Then finally, management considerations. So what we've been using to help guide management decisions is this hierarchy of controls concept. And I credit Sam Gibbs from the US Fish and Wildlife Service for applying this concept. This was originally designed as an occupational health tool to prevent infection of people from hazards, particularly infectious materials, but can also equally be applied to prevent wildlife becoming exposed to SARS-CoV-2 from humans. And it's really a sort of a decision hierarchy, starting with the most effective technique, which is elimination. Obviously, postponing work or avoiding handling of wildlife is going to eliminate that risk. And then next is substitution. Can you substitute what would normally involve handling wildlife for remote monitoring, visual monitoring, or for example, for whiteness syndrome surveillance in bats? We're now collecting fecal samples or guano samples. Then engineering controls using capture methods to minimize distance and contact time with these animals. Administrative controls like washing hands, staying home if you're sick, and then finally the use of PPE, particularly the use of N95 respirators. So I just wanted to show you a case study in the black-footed ferret in terms of how we've been helping management agencies manage these risks. So most of you probably know the black-footed ferret is a very endangered species. Its recovery is dependent on captive breeding. I think over half the population of black-footed ferrets are in captivity. So far, there's been no known exposure of these ferrets, but as I mentioned with mink, a species to which they're related, we do surmise that they're highly susceptible and would potentially suffer from mortality if exposed. In captivity, they're also in close contact with the human caretakers, providing a potential route of transmission. So various actions have been taken to reduce risks to black-footed ferrets, including reducing contact between people and the ferrets, i.e. elimination and substitution, dividing the ferrets into smaller groups with assigned caretakers so infection does get introduced, it would only be transmitted within a small group, so engineering controls. As I mentioned, self-monitoring, regular testing of human caretakers, i.e. administrative controls, and then the use of PPE. And finally, we've also initiated a vaccine trial in a small group of these ferrets to see if actually we can develop a safe and an effective vaccine. So what we did is very simply took a commercially available purified SARS-CoV-2 spike protein. We combined that protein with an adjuvant that's routinely been used in black-footed ferrets that we know that's safe. We initially vaccinated 16 males, and then they received a booster 3 weeks later. We obviously monitored them for any adverse reactions, any clinical signs. We collected serum after the initial dose and then after the boost. And then we ran serological assays, including ELISA and virus neutralization assays, to see if they elicited an immune response to the vaccine. The final phase, which we have not completed, is to do a serum transfer study to assess whether the antibody is actually protective. So rather than doing a challenge study with ferrets, we're actually going to use a proxy species, probably a rodent, that will be transfused with the serum from the ferrets. We can then expose the hamsters with the serum to the virus to see if these antibodies protect the hamsters from infection. And the good news is that there appears to be no adverse effects. They elicited a pretty robust immune response within six weeks of vaccination. This immune response does decline over time, as one expects for most vaccines. And as I mentioned, the serum transfer studies are still pending. So the good news is the experimental vaccine in black-footed ferrets was shown to be safe and immunogenic. We still don't know if it protects these animals against the disease, but so far there has been no infection detected. The titers do decline, so we'll likely need to have some sort of periodic boosting of the vaccine. It's an injectable vaccine. It's useful in captive situations, but not intended for use in the wild black-footed ferrets. But now 2/3 of these captive black-footed ferrets are now vaccinated, and hopefully it's affording a certain level of protection. Then finally, the other activities we've been involved with is a One Health federal interagency 19 coordination team, or sort of interagency coordination. Here in the United States, the CDC has led a multi-agency federal agency response. It's brought together representatives from various key federal agencies from across the federal government, so basically to collaborate to address One Health aspects of COVID-19. There's a zoo and wildlife subgroup that I've been chairing, and we've focused on development of FAQs, messaging, as well as various management guidance documents. And I won't go into a lot of detail on this because I believe Patty Klein is talking about this in more detail. And then finally, in terms of international engagement, and going back to the beginning of the presentations regarding the origins of this pandemic, clearly work needs to focus on how to prevent this happening again in the future. And whilst, as I mentioned, it's not been definitively proven that it's linked to the wildlife trade, it does seem a highly likely possibility. And so, working through the OIE's Working Group on Wildlife, they issued a statement on emerging diseases, emerging zoonotic diseases, and the wildlife trade that called for the better regulation of the wildlife trade, for production of international standards, rather the trade and transportation of wildlife, and development of in-country capacity to conduct surveillance, conduct risk assessments, and manage diseases at the human-wildlife interface. And to their credit, the OIE has gone on to develop a wildlife health management framework. The overall goal is to protect wildlife health to achieve One Health. They have two main objectives. The first is focused really on the wildlife trade, the ability to manage the risk of pathogen emergence at the human-animal ecosystem interface, but then more generally to improve surveillance systems, early detection, notification and management of wildlife diseases, not just related to the wildlife trade. And there'll be 6 main outcomes or outputs as a consequence: improved multi-sectoral coordination and collaboration, increased capacity in wildlife health, better quality data and reporting of these data internationally, the production of guidelines, sanitary standards, risk reduction strategies, improved scientific knowledge, which is disseminated to those that need to act upon that knowledge, and increased public awareness, advocacy regarding these issues. So just to conclude, when faced with a sort of rapidly emerging infectious disease, it's important to deploy multiple techniques to address them, like the COVID-19 pandemic and the nexus with wildlife. Risk assessments, experimental challenge studies, surveillance for natural infections are all very important techniques to assess ongoing risks and fill those knowledge gaps. And simultaneously, it's important to apply a range of management tools to manage those risks, as well as institute developments immediately of tools like vaccines. Interagency and international coordination has a very important role in the development of joint messaging, joint guidance documents, and development of strategic initiatives looking at how to prevent this in the future. So far from this work and other work, obviously the work of many others, it appears that the risks to wildlife from SARS-CoV-2 is low, but continued vigilance is warranted. And so at the National Wild Health Center, we are planning some further experimental trials, as well as some additional targeted surveillance to continue to assess and characterize these risks. So finally, I'd like to acknowledge all the scientists and support staff at the National Wildlife Health Center, particularly Dr. Tony Roque, Dr. Eric Hofmeister, Dr. Jeff Hall, Dr. Honip, Dr. David Blehertz, and many, many others who will obviously contribute to the work I've just presented. They are an incredible, amazing group of folks. So thank you very much for your attention, and if there's any time or ability, I will be happy to take any questions. Thank you.
Hello, I'm Jonna Mazette, and I'm very happy to be with you today to talk about One Health and using that approach to really try to solve emerging health problems, especially emerging pandemic threats and coronaviruses. As we all know, we're incredibly vulnerable. There's on the order of 3 new emerging infectious diseases in people each year. And almost all of those are coming because of our interactions with the planet. And that's why I truly believe that One Health is a critical approach that can really help us move into the future and away from the devastating consequences that we're seeing with COVID-19. Now, can we use this approach to go even further and address what we yet do not know? And so by that, I mean, not just look at COVID-19, look backwards, and not just acknowledge that we're going to have this disease for some time, as well as influenza, but be considerate and aware and ready for anything else that might come. And I do think the One Health approach can help us significantly with that. Before we started the PREDICT project that I'm going to talk to you about, there were about 240 zoonotic viruses known to make humans sick. Because of the work we've done, we've been able to actually estimate that there are probably about 500,000 more or 500,000 more viruses that can infect humans, not necessarily all make us sick, but that should be on the watch list for things that we should be ready for. And I really think that One Health can do that. And for me, One Health has 2 central tenets. One is that it's collaborative, bringing together many different disciplines to address problems in ways that we have never done before. And then the other side is that we're looking at the intersection of what used to be called human, animal, and environmental health. But I think I'm really happy with the progression to pull plants out separately and consider the food systems and food supply separate from the environmental piece. I think that's critical. And so I think if we can bring these pieces together where we're looking holistically and working together, we really have a chance to do so much better than we did with this one and that we have throughout history. Now, if I take that same Venn diagram and list some of the problems that we're dealing with, obviously, as we're all still mostly in sheltering situations and can incredibly connect with the devastating consequences of zoonotic disease, we have to be thinking about those. But loss of biodiversity, I think, is an important topic for us today. And that is another huge piece of this puzzle that One Health can help us address and hopefully help us address together. Now, we'd be remiss if we weren't thinking about the drivers that I've also listed on this slide, just as a few examples. But land use, climate change, economic development, and globalization, all of those things work together to enhance problems, and we need to think about them more holistically to prevent or mitigate the problems that we already have. So about 11 years ago, almost 12 years ago now, a team that I was lucky enough to lead started with the U.S. Agency for International Development, the PREDICT project. And the idea behind the PREDICT project was to find viruses before they spill over, or be ready for them when they do spill over, to be able to mitigate and reduce the epidemic curves. And for us, that meant we really focused heavily on forecasting and readiness and laboratory capacity for early detection. And we were looking in this green area, or what we call the zoonotic pool. So the viruses that were circulating, especially in wildlife, and available to spill over into livestock and people. Now, when they do that, the sort of dotted line up top and the white area, that's where things get out of control. And when we respond too late, we have no chance to shrink those curves. And so the idea behind using One Health to be more proactive is to move all of that response to a more proactive spot and be ready and detect and then respond really early. And that can shrink the amount of virus spilling over from wildlife into livestock, directly into people, or into livestock, allowing the increase and amplification in the livestock and then into people. And the goal, again, is just to reduce those epidemic curves and have many fewer cases and more effective responses. Now, obviously, in COVID, that didn't happen. And we can talk about why. We should talk about why. We should use this lesson to never let it happen again. But we need to build the confidence in science and the political will to act on the science if we're aiming to reduce these curves. So with the project, we went out and worked with more than 30 countries around the world to really start to think about their zoonotic pools, what was circulating in wildlife and livestock and people, but with a heavy emphasis on wildlife, and understand how to safely detect those viruses and be ready for them and work with them in the field and in the lab. And what were we able to achieve? Well, I think, what many people focus on is that we found virus. So we started a project, we sort of built the plane while we were flying it. How do you go out on this large scale and detect and identify not only viruses that you didn't know about before, but also the systems to be safe, and then to use that information to communicate to communities, engage with communities, to prevent the actual risk factors that we documented with behavioral investigations that put people at risk for interacting with and becoming infected with those viruses. So we did sample about 164,000 individuals. Again, most of those wildlife, but many livestock and humans. And we also partnered with FAO and WHO to do that work. And then we got that One Health workforce, almost 7,000 individuals in the countries where we worked, to be doing this in an incredibly safe and informative and collaborative way, and working with more than 60 laboratories in the countries to really bolster their systems to detect any virus that might come their way. Really, that workforce and those labs create what I like to think of as almost a global immune system. They not only help us to identify threats before we're experiencing them, but to jump in quickly and put in the risk mitigation measures that help us control, which didn't happen on this one and need to be addressed for the future. So what about coronaviruses? So I mentioned we found about more than 900 novel viruses, about almost 300 known bad actors, known viruses using the same platform. I think that's important as well. So these labs can detect knowns and novels. When we detected the knowns, we were also adding to the rich information about important viruses that make people sick, and identifying new hosts and identifying new locations where people weren't looking for those viruses beforehand. But I think if for our conversation today, identifying about 177 coronaviruses, more than 160 of those brand new coronaviruses to science, helped us to really identify host ranges, how their binding, receptor binding is working, where we should be looking for them, what kind of interfaces. And that helped a lot in the countries where we were working, absolutely, with COVID-19. They jumped into action. Their labs were ready. They responded immediately, and some of them had the best outcomes of many countries around the world. It also helped us with the science to be able to look for where this might go and how we can control it in the long term. I am a veterinarian. I'm an epidemiologist with a veterinary background, and I think it's interesting to note that coronaviruses have been very important veterinary pathogens for decades. So while there are 7 known human coronaviruses, for more than 30 years before we knew about human coronaviruses, the veterinary community was dealing with coronaviruses that affect the food supply, frankly, also our pets. And so that's just another example of this power that if we're working together, we could be looking more completely at the complexity of what's happening in our natural environment and preparing for the future. In the PREDICT project itself, one of the first things that we had as an early important discovery around coronaviruses, in addition to finding new ones and starting to map them and understanding regional differences and host differences, we were able to actually identify that rhinophilid bats were really important with SARS and SARS-related coronaviruses, and start to think about and identify the ACE2 receptor as showing that actually the viruses didn't necessarily have to go through an intermediate host. They could spill over directly from bats, and obviously spill over also into intermediate hosts and be available even more broadly to people. So we got out there, we looked at a lot of animals, primarily non-human primates, rodents, and bats, and the bats were definitely the richest source of our discovery of coronaviruses. And I think one really important thing for discussing today is that we were able to identify different classes and variants within the SARS viruses or the coronaviruses, and including the SARS-related viruses, and the SARS coronaviruses here were predominantly infecting and being detected in rhinophilid and hippocytarid bats. That was incredibly helpful because we could say, look, the most likely suspects of where we should be looking for the risks. And again, the risks that people put themselves in. The bats are not leaping out and making people sick with their viruses, and the viruses are also not available to just attack us. It is how humans, we are behaving with those bats and with the environment that are putting us at risk. But understanding their hosts helps us to start understanding where we might be looking and enhancing surveillance in humans and be ready for these viruses. And here's an example of that. If you look at the distribution of bats that harbor where we detected SARS-related coronaviruses, you can see that it is not just China, and that we should really specifically, if you look at the Rhinophilus affinis map here, we should be looking at all of Southeast Asia. And that's proving to be correct as we are detecting more and more and going into archive samples and things and finding more SARS-related coronaviruses that all help us build the picture of what happened here and the likely evolution of SARS-related coronaviruses. We're also able to start to identify other coronaviruses, likely evolutionary strains of important coronaviruses like MERS-like coronaviruses, and even identify cryptic biodiversity, with this bat actually being new to science when we found a MERS-like coronavirus in it. And we're able to start to look at this bat and its most closely related cousins that were filling the same niche and start to look at their distribution and where maybe we should look for the evolutionary source of MERS and how that overlaps with camels and camel breeding and all of that good information. Also, we could look at the risk factors. So we were doing a lot of work with humans, behavioral surveys, serosurveys, testing them for viruses in the same interfaces as animals. And, the large market hypothesis and concern has been very important. And you can see here that when our teams in Vietnam were sampling in the field at the same interfaces where trappers and traders were collecting rodents to take them either to farms or to large markets, we're seeing about 20% of those rodents infected with coronaviruses. Then, of course, with the intense husbandry, stacking up and mixing at the market, we saw that grow to over 30%. And then most concerningly, by the time that those same rodents were getting to the actual end user, the restaurant and the consumer, we saw that they were infected more than 50% of the time, making coronavirus available to those clients in that restaurant interface. So we're able to really start to map these risky behaviors that can inform mitigation, behavior change, and all of those interventions that we need to keep this from ever happening again. In addition, we're able to start to pull together risk factors, not only virologic, which are critically important, but also risk factors including the ecological and epidemiological occurrence of hosts and contact with humans, and put that all into a web interface there where people can enter their own new viruses and sort of crowdsource this information, but also to rank them for riskiness, but also start to rank and make that watch list for increased surveillance for the ones that appear to be most risky. And if I just pull up here an output from that application for coronaviruses, you can see that SARS-CoV-2 is at the top of the coronavirus risk list for spillover, which has some indications for spread as well, and that one of our predict viruses ranked very highly as well. We call it Predict 35, but it down at the bottom of your screen. And this looks like a credit score because we are looking at multiple factors that include host factors, environmental factors, as well as virus factors. Finally, I just want to make sure that we all take this message out to all of our networks and colleagues that we need to protect bats. Bats are critical for ecosystem services, protecting biodiversity. We need them. They're pollinators, they're seed dispersers. They also are insectivorous and protect our crops. And attempts to reduce bat populations are generally unsuccessful. They encourage recruitment, making more bats. They also have been shown to increase the shedding by stressing the population. So we need to protect bats. And to help do that, we put together community outreach materials, including a book that's designed in 2 different forms for Africans and for Asia, Living Safely with Bats. And it's been translated into more than 12 languages now, so I'm thrilled about that. So thank you for letting me share with you today. I look forward to the other presenters and this important session. So take care.
Hello, everyone. My name is Tom DiLiberto, and today Dr. Susan Schreiner and I will provide you an overview of the SARS-CoV-2 outbreaks in U.S. farm mink and the wildlife epidemiological investigations we participated in as part of the One Health responses resulting from those events. While humans are the primary hosts for SARS-CoV-2, there have been hundreds of spillover events into various animal species worldwide, the most significant of which are infections in farmed mink, with over 400 farms in 11 countries reporting infections. The U.S. has reported about 59% of the confirmed positive cases in animals, with most detections occurring in dogs, cats, and farmed mink, followed by zoo and other captive wildlife, and of course the one wild mink that we reported in December. With respect to farm mink, the initial outbreaks were reported in Europe, with the most cases in Denmark, Poland, the Netherlands, Finland, Lithuania, and Greece. Europe has a much higher density of mink farms than the US, and the response in most countries there has primarily been culling of infected farms. Europe also has molecular epidemiological evidence that demonstrates transmission from humans to mink and from mink back to humans. Such reverse zoonoses has not been documented in the US. Global mink farm outbreaks peaked in the fall prior to the pelting season in December and January. Of the 416 infected mink farms, 16 have been in the US. Mink production in the US is primarily through small family farms spread throughout the country. Production has been falling over the last several years, associated with a reduction in the price of pelts, but there were still 2.7 million pelts produced in the United States during 2019. Wisconsin, Utah, Idaho, and Oregon remain the top producing mink states. There have been 4 states that have experienced SARS-CoV-2 outbreaks in farmed mink. These are Wisconsin, Utah, Michigan, and Oregon. And as we saw in the previous slide, 3 of these, Wisconsin, Utah, and Oregon, are in the top 5 mink producing states. Preliminary analysis indicates that all these outbreaks are associated with human to mink transmission by infected farm workers. Biosecurity on US mink farms is quite variable. Not all farms have perimeter fences around sheds to contain escapees, and those that do may or may not have features to minimize movements of wildlife on and off the properties, or to decrease the attractiveness of the farm environment to wildlife. Mink themselves are highly susceptible to SARS-CoV-2 infection, and once infected can be asymptomatic, exhibit only mild clinical signs, or experience high morbidity and acute mortality. Consequently, it's extremely important that extra precautions are taken to protect mink as well as humans working on the farms. Detections of outbreaks on mink farms in the US has relied primarily on morbidity and mortality surveillance by owners. If an unusual level of morbidity or mortality was observed, the owner could contact a veterinarian and samples sent to the state diagnostic lab. Non-negative samples were forwarded to the USDA National Veterinary Services Laboratory in Ames, Iowa for confirmatory testing. If positive, state and federal agencies initiated a One Health epidemiological investigation on and around the farm in question. It is currently unknown if or how many mink farms have gone unreported through this surveillance approach. And USDA is currently working with the state departments of agriculture and the industry to develop active surveillance plans for mink farms during the upcoming breeding season. Outbreak response of SARS-CoV-2 on U.S. mink farms has been through a coordinated One Health effort, including the CDC Special Pathogens Branch, USDA Veterinary and Wildlife Services, the USGS, and state natural resources, agriculture, and public health agencies. The CDC has been sampling the farmed mink and domestic animals on the farms, as well as conducting the human epidemiological investigations with state departments of health. USDA Wildlife Services, through its National Wildlife Disease Program and National Wildlife Research Center, has been conducting wildlife sampling in collaboration with state departments of natural resources and agriculture. Wildlife testing of samples has been conducted by USDA National Veterinary Services Laboratories and the USGS National Wildlife Health Center. With respect to the wildlife response, the challenge has been where to focus very limited resources, given that there was virtually no information on susceptibility of wildlife species found on and around those farm environments. On-farm investigations have focused on whether SARS-CoV-2 escaped the sheds and infected free-ranging species. And if so, was there evidence that the virus was circulating in wildlife populations? Wildlife sampling was primarily conducted using cage traps and focused on mesocarnivores. However, small mammals were also targeted on 2 of the first set of positive farms identified in Utah. When feral cats were captured in cage traps, attempts were made to sample and release those animals. We conducted wildlife sampling on 8 of the 16 positive farms in all 4 states. In response to the initial outbreak during August in Utah, all escaped mink and wildlife species captured were euthanized, and necropsies were conducted to collect nasal, oral, and rectal swab samples, tissue samples, and serum for testing. Results from those initial investigations indicated that all confirmed detections were identified through PCR testing of swabs or virus neutralization tests on serum. Therefore, in subsequent outbreaks in Utah, Michigan, Wisconsin, and Oregon, animals were euthanized and only swab and serum samples were collected. Today, we'll share with you the preliminary highlights of the wildlife sampling conducted in all 4 states. Across all farms, we captured just over 250 mammals, including mesocarnivores, rodents, and lagomorphs. Of those, we only confirmed positive detections in mink. We captured 21 mink, 17 of which were escaped farmed mink in Utah and Oregon, and 4 were wild mink in Utah and Wisconsin. Note that we did not capture any mink associated with the single positive farm in Michigan. Of the 17 escaped mink, 16 were positive by PCR, serology, or both. Only 1 of the wild mink was positive, and it was positive by PCR. We also recovered a virus from that individual. While sequence analyses are in progress, preliminary results indicate that all 3 viruses we recovered from escaped and wild mink are identical to those found in the farmed mink. Now, as we indicated in our OIE report in the ProMed Mail post on December 12th, the wild mink we found positive was the first known case of a free-ranging native species confirmed with SARS-CoV-2. That wild mink was captured in a natural creek habitat 672 meters from an infected shed in Utah. In this diagram, the red dot labeled A is the infected shed, and the purple dot labeled C is the capture location of the wild mink. The green dot labeled B is the location of an open pit where carcasses of dead infected farmed mink were being deposited by the owner. That pit was about 70 meters from the creek and only 242 meters from the capture site. The yellow dot represents the point at which the creek is closest to the sheds, about 578 meters. Given that the mean home range size of a wild mink is about 5 square kilometers, the mink could have been exposed to SARS-CoV-2 by accessing the farm site directly from the creek or by contact with infected carcasses in the pit. An interesting side note to this story is the challenge we had in determining whether the mink we captured were wild or recently escaped farm mink. Given that farm mink are the same species, Neovison vison, distinguishing the two is not straightforward. Genetic analyses of individuals to classify the animals as escaped versus wild was of limited value because an adequate genetic reference library for wild mink in the U.S. does not currently exist. Also, studies in Canada, such as this recent one by Bowman et al., found that farm mink regularly escape, and escapees can hybridize with wild mink, complicating wild versus farmed identification based solely on genetics. However, we were able to conduct microsatellite analyses to determine if a captured mink was genetically similar or distinct from farmed mink sampled in the area. We identified 8 genetic clusters, many shared between the single farm in Oregon on the top and the 4 farms in Utah on the bottom. Here, each rectangle represents a sample for an individual mink. The colors represent genetic clusters, and the letters represent the 4 farms in Utah. The wild mink that was positive for SARS-CoV-2 is represented by the dark green rectangle at the bottom right in this diagram. Notice that it does not cluster with any of the farmed mink samples tested. Also notice that the 2 dark brown rectangles just to the left of the dark green one represents our 2 other wildlife mink samples from Utah. While these 2 wild mink samples clustered with 1 sample from a mink in Farm B, we still called these mink wild. That's because given the limited value of genetic analyses and the propensity for some escaped mink to hybridize with wild mink, we relied more heavily on other characteristics in deciding if a captured mink was wild or escaped. For example, was the animal caught on or off the farm? Was it in native habitat or in a farmed or other human-altered habitat? Was it morphologically similar to farmed or wild mink? Wild mink are typically milk chocolate brown, whereas farmed mink are black, white, or silver. Also, wild mink have prominent guard hairs, as you can see on the top animal here, while farm mink have few guard hairs, such as the black animal on the bottom. Additionally, live-trapped escaped mink tend to exhibit relatively docile behavior as opposed to wild mink, which are aggressive in traps. And if necropsies are performed, physiological differences can sometimes be observed, such as the presence of large amounts of subcutaneous fat and even fatty livers in escaped mink, which are rarely seen in wild mink. Given the relatively focused trapping efforts on and within a 3-kilometer buffer of infected farms, evidence to date suggests that although the SARS-CoV-2 virus escaped the sheds on some of the infected mink farms, there is currently no evidence that the virus has become established in wildlife populations. Furthermore, given that the viruses identified in farmed, escaped, and the wild mink were identical, it is unlikely that the one isolated from the wild mink passed through an intermediate host of another species. This suggests that the wild mink likely contracted the virus either through direct contact with an infected farm or escaped mink, or through indirect contact with infectious material such as a carcass, feces, or other attractants. It's also important to remember that currently the SARS-CoV-2 virus is a human-adapted pathogen. We know that SARS-CoV-2 infection spreads most commonly from person to person. Infected people, either with or without symptoms, can introduce the virus to farmed mink. Once introduced into a shed, infected mink rapidly spread the virus to other mink, resulting in mortality rates from essentially 0 to over 40%. We're still learning about how long mink are infected with the virus and how it can spread to other animals on the farm, like dogs, cats, escaped mink, and wildlife. And finally, At this time, there's no evidence to suggest mink to human transmission has occurred on U.S. farms, as was observed in Europe. State Departments of Agriculture and Health, the CDC, and USDA are working with farmers to increase awareness of the potential for workers to infect mink, to heighten the biosecurity efforts on farms, and to develop active surveillance plans for the upcoming breeding season. While currently there are no actively infected farms in the U.S., if additional infections are identified, wildlife surveillance will be included as part of the One Health investigations. Also, experimental infection studies are being conducted to determine which species are at greatest risk of infection. A recently posted preprint paper by Colorado State University and the National Wildlife Research Center found that deer mice, bushy-tailed wood rats, and striped skunks were susceptible to experimental infection with SARS-CoV-2 and shed the virus in their respiratory secretions. Alternatively, cottontail rabbits, fox squirrels, Wyoming ground squirrels, black-tailed prairie dogs, house mice, and raccoons were not susceptible to infection. Finally, we wish to thank all our state and federal partners for their participation in these One Health investigations, especially to those landowners who gave us permission to trap on their properties. Thank you very much.
Good morning. Thank you to the moderators and to the organizers for letting me speak this morning. My name is Jonathan Cook. I'm a postdoctoral researcher at Patuxent Wildlife Research Center, and today I'll be talking about some work that myself and my co-authors have been doing to develop rapid decision-making tools to evaluate the human-to-bat SARS-CoV-2 spillover risk. The fundamental question driving this research is whether or not North American bats are at risk of SARS-CoV-2 spillover from humans to bats. And part of what stimulates that is that we think the horseshoe bat may have played a role in COVID-19 spillover to humans in the first place. And so what we've attempted to do is to capture the diversity of North American bats by doing risk assessments for the little brown bat, the big brown bat, and the free-tailed bat. One reason that this work is particularly urgent is because of the time-sensitive conservation work that's ongoing with Pseudogymnoascus destructans and localized populations of bat species. It's really important to be able to track the spread of white nose syndrome to assess the near-term risk to localized populations, but to also project into the future the long-term impact of Pseudogymnoascus destructans and white nose syndrome infection, and also to continue to conduct in situ research on white nose syndrome intervention tools. When you put together the unknown risk of SARS-CoV-2 spillover to North American bats with the need to continue time-sensitive conservation work, suddenly pretty simple questions about field work become quite difficult. And so what I'm going to argue is that performing rapid risk assessments guided by decision theory can really help clear up that complex decision context by framing the decision around key objectives that a managing agency is trying to accomplish by developing an analytical approach driven by various types of data, and I'll talk about how we did that for SARS-CoV-2, and then to produce results and identify key unknowns that are particular to the disease system. Those results speak directly to the objectives, and therefore they're able to inform decisions and direct policy and research, and finally lead to knowledge gaps where the scientific community can jump in and resolve some of these issues, hopefully, and we can revisit that decision in instances where that decision is necessary on a recurring basis. Now, the first rapid risk assessment that was done in March 2020 relied on the steps of decision theory that I just discussed. And as a result of that structured approach, it brought together a broad stakeholder group with diverse needs and backgrounds. It clearly identified management objectives, things like the need to continue to conduct conservation programs, but also mitigate the risk of SARS-CoV-2 spillover. It identified and evaluated management mitigation efforts, such as the use of personal protective equipment, and it clearly identified sources of uncertainty. The result was that the authors determined a small but non-zero risk of zoonotic transmission as a result of human interaction with North American bats. And ultimately, it provided peer-reviewed science that could be used to inform decisions and policies by speaking directly to what's relevant to a managing agency. And it was used by a variety of agencies in their policy directives and their decision-making throughout the spring and the summer. However, one large uncertainty remained after that risk assessment, and that's how susceptible bats are. I mean, it's sort of a fundamental question to being able to perform a risk assessment, but there was very limited information to inform an estimate at the time. And so as a result, the expert panel that was engaged in this process estimated that there was anywhere from a 0% chance all the way up to a 100% chance that bats were susceptible to SARS-CoV-2. And that's not really surprising given all the uncertainty surrounding COVID-19 at the time. If you think back to what we knew in March, it was very limited. And so fast forward through those spring and summer months, and we start to be confronted with a slightly different question, and that's what is the risk of zoonotic transmission during winter field work, which is slightly different. It requires researchers to enter fairly enclosed spaces with bats in close proximity, and so we might think or hypothesize that the risk is a little bit different. And so we engaged in this process with a steering committee to evaluate that risk for decision makers during the winter field season. And so I mentioned earlier that one of the benefits of this structured decision approach is that we can create new knowledge and re-examine decision based on that new knowledge. And for SARS-CoV-2 risk, we had several lines of new evidence. We had bat challenge studies that were completed that gave us some insight about the susceptibility of species. We learned a little bit more about how the virus binds to cells and leads to infection. We also learned a lot in the human-to-human transmission realm about the importance of aerosolized transmission, and so we can translate some of those findings into our bat work. And finally, we learned about the survival of the virus outside the host on surfaces, and we can translate that to surfaces within the cave to understand a little bit about the contact or contaminated surface exposure that bats might be experiencing. And so what we did was we put together, again, a steering committee, and we looked through the scientific literature, the advances that have been made, and we developed an infection risk model with 2 transmission pathways that were really relevant to the decision of whether or not field work in the winter should occur and how risky it was. The first pathway was aerosolized exposure during activities in enclosed spaces with bats. So we're talking all of the bats within a hibernacula or roost site here. And also the risk from a bat contacting a contaminated surface within that hibernacula or roost site. And the second pathway digs a little bit deeper into the activities that humans are doing with bats, and that's looking at the aerosolized exposure for bats during handling activities. So as researchers are picking up bats, evaluating their condition, and coming into close contact with them and potentially emitting viral particles that the bat can be exposed to. We also considered the deposition of viral particles on the fur, the face area of the bat itself. So that's sort of all wrapped up in pathway 2. In a conceptual model, what we did was we broke down aerosolized transmission into 3 distinct steps, and we quantified each one of those steps based on the available literature. Starting with emission of virus by humans, there's actually quite a bit of studies that give us values that we can use to inform how much virus is being emitted by humans. For bat exposure, we were able to rely on some developed physical models that help us to understand how emitted virus accumulates in space, homogenizes in an air column, and leads to exposure. We were also able to consider the specifics of the cave, the relative humidity, the temperature, the volume of the cave, as well as the respiration rate of particular bat species to get us a good estimate of the magnitude of exposure. And then I mentioned that during the first risk assessment, there were very little data to inform the susceptibility of specific species to SARS-CoV-2 given an exposure event. And so what we did was we circled back to this question with another expert panel made up of 12 experts. And we asked the panel to use and integrate the new information that was available and give us an updated estimate on the susceptibility of bats based on the challenge studies and some other information that was available to them. And when we revisited this question, experts estimated that bats were quite a bit less susceptible than we initially estimated in the March risk assessment. And so we incorporated this new information into that updated winter risk model. So based on the updated infection risk model, we can circle back to the questions that were quite challenging initially without sort of this mechanistic model and strategic use of empirical and expert elicited data to get at the mechanistic relationships between human activities, cave conditions, bat physiology, and the ultimate risk of SARS-CoV-2. Digging into our specific results, I'm going to speak first to the question of should fieldwork occur at all. And our thinking here was that it's important to evaluate the risk of entering a cave and surveying animals as a function of cave condition, but also the localized prevalence of COVID-19 in the human population. So really trying to get a handle on sort of that first cut of that decision of whether or not we should enter the cave at all. And so what we did was evaluated the species-specific infection risk as a function of cave volume and COVID-19 prevalence as sort of examples of the ways we might think about this question. And what we found, I'm going to talk about the results for the Mexican free-tailed bat because it turns out that our expert panel estimated that species to be at the highest risk of being susceptible to SARS-CoV-2. And what you're seeing here on the y-axis is the probability that greater than one bat is infected during any one field survey, and that's in a percentage. And on the x-axis is the cave volumes, the range of cave volumes that we explored to sort of quantify that risk of infection. And what we can see is that at smaller cave sizes, there's about a 0.3% of risk that greater than one bat is infected during any fieldwork excursion. And that risk declines non-linearly through the different cave volumes that we explored all the way up to 3,000 meters cubed. And we can do the same thing for the other species we explored, the little brown bat and the big brown bat, and put specific risk values on those species based on those localized cave conditions. We also evaluated the question as a function of COVID-19 prevalence and found a positive increasing relationship between COVID prevalences of 0 and about 5%. And the probability that greater than one bat was infected ranged from 0 up to just under 0.1%. And so next, we dug one layer deeper to look at, okay, so we're entering the cave, what's that sort of risk look like? And we're digging in to evaluate how risky is it to actually pick up a bat and handle that bat, given that second pathway that we looked at. And so what we did here was we evaluated the species-specific infection risk as a function of number of bats handled. And we picked little brown bat to display here today because that's typically one of the species that we're handling to do this white nose syndrome work. And what we found on the x-axis, we looked across 0 to 50 handled bats, which is pretty typical for field work. And on the y-axis is quite a bit smaller magnitude than those previous figures I showed, with the probability that greater than one bat is infected from handling ranging from about 0 up to 0.005%. So pretty low risk here. And not surprisingly, when we look at that handling risk in the larger context of the infection risk model, we can see that it doesn't overall elevate the risk of infection for the bats within the hibernacula, but that it does slightly increase the risk as we're handling bats. And part of what goes into that is that we're handling bats for a short period of time, and so that viral emission is actually quite low. And we also looked at a baseline scenario where there is no white nose syndrome within the hibernacula, and a scenario where there is white nose, and that's that thinner solid line on the top of the figure there. And what we found was that risks were slightly higher for white nose infected hibernacula, just because of that co-infection and the immune depression that happens with active white nose syndrome within a hibernacula. And so addressing the question of whether or not risks can be mitigated, what we found was actually that masks, in our case we evaluated surgical masks, and pre-survey COVID-19 testing was quite effective. So if you remember back to the cave volume figure that I just showed, we can see that this is the line that I described as being sort of the baseline risk, and we can see that here testing is reducing that risk quite substantially, as is masks, this dotted line below that. It's a similar story for COVID-19 prevalence for both the masks and the pre-survey testing. And finally, for the handling, these two lines down here show testing, the effect of COVID-19 testing for both caves that have white nose and without white nose, so quite substantial reductions from those baseline conditions. So I hope what I've shown today is that rapid risk assessments guided by decision theory can be a powerful approach to produce reliable results quickly, to estimate risk given available data and knowledge, and here experts are really key to driving that process, to identify effectiveness of mitigation measures, to identify key unknowns for future research, and to incorporate new knowledge to improve understanding and estimation. And furthermore, these processes can happen quite rapidly. For the first risk assessment, we completed the work, Dr. Runge completed the work in just around 2 weeks. For the second risk assessment that I was a part of, it took us about 6 weeks to get really specific results in the hands of decision makers. And with that, I'd like to acknowledge the expert panel that were critical in this work, as well as the steering committee and many others that I haven't listed here. Thank you very much.
Thank you for the introduction and the opportunity to discuss our work with the SARS-CoV-2 virus and white-tailed deer. All of the work I'm going to present today has been a collaboration between researchers at the USDA's National Animal Disease Center in Ames, Iowa, and Dr. Diego Diel and colleagues at the Animal Health Diagnostic Center at Cornell University College of Veterinary Medicine in Ithaca, New York. An analysis of 335 emerging disease events occurring between 1940 and 2004 showed that more than 60% were zoonotic, and over 70% of those had an epidemiologically important wildlife host. One of the worst case scenarios would be an emerging zoonotic disease agent of livestock, poultry, or wildlife that jumps from its animal host to humans and achieves efficient human-to-human transmission, that is, achieves pandemic potential, causing high rates of mortality, resulting in devastating impact on public health and possibly livestock, poultry, or wildlife. We are today in the midst of one of the most devastating pandemics in this century, caused by a zoonotic agent that has been transmitted from an unknown wildlife species to humans. Therefore, it is critical to understand which animals are susceptible to infection with SARS-CoV-2 and which animals are capable of transmitting the disease to humans or other animals. Others, including us at the National Animal Disease Center, have examined the susceptibility of poultry, cattle, and pigs. Thus far, studies have demonstrated no susceptibility or limited susceptibility. But why did we go on to look at white-tailed deer? First, understand that the SARS-CoV-2 virus uses as a receptor on cells a protein known as ACE2. Once the virus binds to the ACE2 receptor, it is internalized and the cell is then infected. Last year, in a paper by DeMoss in the Proceedings of the National Academy of Sciences, they examined the ACE2 receptor binding region of various animal species, comparing them to the human and predicting the potential for binding with the SARS-CoV-2 spike protein. They then categorized the binding potential for each species from very high to very low. Not surprising, many species of non-human primates fell in the very high probability grouping. Somewhat surprising, however, 3 species of deer, white-tailed deer, reindeer, and paired David's deer, fell in the high probability grouping. Given all these considerations, we chose to evaluate the susceptibility of white-tailed deer to experimental infection with SARS-CoV-2. We had 6 2-month-old fawns remaining from our breeding herd to use in this study. Using an isolate that our Cornell collaborators had isolated from a tiger in the Bronx Zoo and then had traced that back to the tiger's caretaker, we intranasally inoculated 4 fawns, using the remaining 2 fawns as non-inoculated contact sentinels. Baseline temperatures and pre-inoculation samples were obtained. After inoculation, body temperatures were evaluated daily using subcutaneous chips and a chip reader. Nasal and rectal swabs were collected for PCR to detect viral RNA and virus isolation to detect replicating virus. This was done daily for the first 7 days and then on days 10, 12, 14, and 21. Blood was collected for virus isolation and serology for antibodies to SARS-CoV-2 on days 0, 7, 14, and 21. PCR, virus isolation, and serology were performed by our collaborators at Cornell. It is worth describing the room in our biosafety level 3 facility in which the fawns were kept. The room was approximately 12 feet by 12 feet, with a barrier approximately 4 feet high dividing the room in half to prevent nose-to-nose contact between inoculated and sentinel deer. Airflow inside these rooms is kept at 10 to 11 changes per hour, and the direction of the airflow moved in a manner that was primarily from contact fawns towards inoculated fawns. During the 21-day study period, no fever was detected, and there was no change in behavior or appetite. If we look at PCR results of nasal swabs, now PCR results are detecting RNA, viral RNA, and not necessarily replicating virus. These first 4 graphs are inoculated fawns, and we can see that in their nasal secretions, they shed viral RNA, most of them for the 21-day period of the study. We also note, however, in the contact sentinel fawns, these bottom 2 graphs, that they began shedding viral RNA by day 2. These bars represent viral RNA from rectal swabs, and we can see that all fawns, inoculated and contact fawns, shed viral RNA intermittently in their feces. If we look at virus isolation results now on the right, which look at infectious replicating virus, not just the RNA, we see that virus was shed for about a 5 to 7 day period. And in contrast, viral RNA was found on rectal swabs only on day 1, 24 hours after inoculation, probably the result of pass-through from swallowing some inoculum. And this probably suggests that the GI tract is not a site of replication for this virus. Following post-mortem examination on day 21, it was revealed that multiple tissues from all fawns were PCR positive, contained viral RNA, but virus isolation negative, meaning there was no replicating virus present. In all of the fawns, the palatine, tonsil, and medial retropharyngeal lymph nodes were PCR positive. Other tissues commonly positive were other lymph nodes of the head, such as the submandibular lymph node and the nasal turbinates. Other random scattered tissues were also positive by PCR. However, note that the lung was negative for viral RNA. Also important, all fawns developed high levels of virus neutralizing antibody by day 7, And we can see that antibody titers in inoculated fawns in blue were generally higher than those seen in the contact fawns in red and brown, which could very well be a dose effect, and that the dose that the contact fawns were exposed to was likely somewhat less than the dose given to inoculated fawns. Our conclusions from this study are that unlike other ruminants, such as cattle, white-tailed deer are susceptible to experimental infection with SARS-CoV-2, but infection and viral replication do not result in clinical signs. SARS-CoV-2 replicates and can be shed in nasal secretions for at least 7 days after infection. Fecal shedding and replicating virus appears to be minimal. Infected deer develop virus-neutralizing antibodies by day 7. Viral shedding is of significant magnitude to transmit virus to other deer. Some other conclusions that we noted. We did not find virus in the blood, so viremia does not seem to be a feature of this experimental infection model. And viral RNA can persist in lymphoid tissues for at least 21 days, although replicating virus is cleared much earlier. So the question comes up, how did the contacts become infected? We're confident that there was no nose-to-nose contact. However, virus could have gone over or under the barrier in water or water droplets as the pens were cleaned. It's also possible that true aerosol transmission occurred with virus-laden droplets that can remain suspended in the air and carried over long distances. So that summarized our first study in white-tailed deer, and a preprint of that paper is available on bioRxiv online, and it's been submitted to a scientific peer-reviewed journal. We just completed our second deer study, and this is the first time I've presented this data as it's just coming in, and so I don't have all of the results yet. In our second study, we wanted to confirm our findings in study number 1, that being that deer are indeed susceptible to infection, but we wanted to better establish the shedding patterns. We wanted to better control possible routes of transmission and eliminate some of the unknowns of the first study. So there were a couple of differences in this study. First of all, we used deer that were approximately 8 to 9 months old. These deer were inoculated with an isolate similar to the most prevalent human isolate circulating in the U.S. rather than the Tyget isolate. So similar to before, no fevers were recorded during the 21-day period of the study, but some deer did have mild mucus discharge from the nose. One deer was noted coughing several times, and there was one deer that for several days in a row, after handling, would move to a corner and lie down for several minutes. None of these signs were severe, and all were categorized as mild. It might be helpful if I explain how we examined deer-to-deer transmission in study number 2. First of all personnel wore personal protective equipment to prevent not only zoonotic transmission from deer to us, but also reverse zoonotic transmission, potential transmission from us back to the deer. This given their apparent high susceptibility to infection with SARS-CoV-2. In study number 2, we had 3 negative control deer that were never infected, that remained in a single room the whole time. Dedicated staff cared for, fed, and collected samples from the negative controls. These staff members never did anything with infected deer. 6 deer were intranasally inoculated on day 0. On day 3, 2 clean deer were moved to a clean room. The inoculated deer were then introduced, and these deer commingled for 48 hours. The 2 contact deer were bled and oral nasal swabs collected on entering the room and leaving the room 48 hours later. These 2 contact deer were moved out and kept together in their own clean room. When the inoculated deer left their room on day 3, it was decontaminated twice, the feed was discarded, and let sit for the 48-hour period. On day 6, 2 clean deer were moved into that clean room with clean feed, and the inoculated deer were moved back and introduced, and these deer commingled for 48 hours. Later, the 2 contact deer were moved back to their clean room. We took these precautions because we wanted to be sure that contact deer were becoming infected from the infected deer and not from contaminated surroundings where the inoculated deer had been housed or the contaminated feed the inoculated deer had been eating. So here are the results so far, and we only have PCR results. We don't have any data on virus isolation and infectious virus. Similar to study number 1, most inoculated deer shed viral RNA in their nasal secretions for the duration of the study. The other lines that you see here paralleling the nasal secretions are the saliva. So viral RNA was shed in both saliva and nasal secretions in a similar pattern. We also see, similar to study number 1, intermittent shedding of a low amount of viral RNA in the feces. The 2 contact deer that began commingling with inoculated deer on day 3 had viral RNA in their oral nasal secretions on day 5 when they were removed. No viral RNA was detected in deer commingling from day 6 to 8. And 1 fawn that commingled on days 9 to 11 had a low amount of viral RNA in its oral nasal secretions. The other was negative. At postmortem examination on day 21, 3 of 6 inoculated fawns and 1 contact fawn from each period had very mild lesions limited to the right cranial lung lobe. These deer also had variable amounts of mucoid discharge in the trachea and the bronchi. So similar to study number 1, we found that deer were susceptible to infection, this time with a different isolate. Deer shed viral RNA in their nasal secretions and feces. Deer transmitted viral RNA to non-infected deer. Things we saw in study number 2 that we did not see in study number 1 were mild clinical signs, again not enough to alter their appetite. We saw mild gross lesions this time, and we detected viral RNA in saliva, which was a sample that we didn't collect in study number 1. We're still waiting on virus isolation results, serology, and histopathology from this second study. In the future, we would like to look at mature deer to see if there is any age-related difference in susceptibility. We would like to conduct time course studies to characterize the pathogenesis. We would like to conduct dose titration studies to determine the minimum infectious dose and determine if after infection, deer can become reinfected, or does infection confer immunity? Thank you for the opportunity to share these results, and I'll be happy to take any questions.